Hello, everyone. Welcome to the third session of the Opportunities in Bioengineering. And today we will be discussing the prospects of engineering in manipulating cells and tissue and how they may be impacting the organisms. So today, the two panelists who are joining me are Dr. Kapish Gupta, who is a postdoctoral researcher at the University of Pennsylvania. And
Hello.
hi, Kapish. And the second panelist who joins me today is Dr. Nilesh Kumar Dubey. He's
Hello.
Hi, Nilesh. Yeah,
Hi.
so Nilesh is a clinician turned biomaterials researcher in the University of Michigan, and both of them have been doing quite a bit of research in tissue engineering for almost like 5, 6, or 7 years, maybe 6, 7 years, something like that. Anyway, both of them began in India. Good for, interesting thing is one of them began as an engineer, another one began as a dentist, and now both of them are working on different aspects of the same thing, which is quite interesting. That most of the people, even though they begin at different places, they could end up at the same place, uh, and that's very similar to the theme that we are following so far. That there are some people who have been pure biologists, some biophysicists ended up doing a similar kind of thing. Somebody who was a biotechnologist, another biomedical engineer ended up doing similar kind of thing. Now the two of you, that one is a biotechnologist from the engineering side and another one clinician, and ending up doing similar kind of thing, not the same, of course. We will realize soon how they are different from each other. So yeah, going back to tissue engineering, my first encounter I would like to share with you with tissue engineering, or when I got familiar with the idea of tissue engineering, was when I got to read about the green fluorescent mouse. I'm not sure if it still exists in the NCRT book or not, but that's how I got acquainted with this entire organism, which could be changed just by tweaking a gene. And then the other idea that I got acquainted with was how you can treat, or how it was possible to treat. Not it could not be treated yet very well, but how it was possible to treat severe combined immunodeficiency disease just by stem cell therapy, so by injecting a cell. And that's still a far-fetched idea right now, I think. And I think Nilesh has something to talk about it more.
No, it's like a very interesting field, Like before, it used to be a science fiction. We used
Right.
to watch in movie, but when we come in research, we realize, this is no more science fiction. And I got introduced with this idea first when my brother-in-law, who was working in WHO, and
Huh.
I, being a dentist, at that was time I was in my third year, and I wanted to do a research. My father is also a researcher, okay, but he's more from a rocket scientist.
Ah,
Then
okay.
I had this thing of, okay, what research really means. Like back home, if you're a clinician, you just do clinics. What is research? You don't care much. And I had
Yeah.
that idea. And my brother-in-law, he told me, he's working on liver regeneration and lung regeneration. I was like, how you do that? And that was the first point for me, and that really excited me to make up my mind in third year of my dentistry
Right.
that I will go for a research and try to do a clinician. And I got introduced firstly when I came to MD Anderson in 2010. And I
Right.
worked with liver cancer actually, and
Ah.
we tried to
Oh.
regenerate those tissues. So that
Yeah.
was my first encounter, and that's really made me. And I pursued PhD from NUS. Now I'm in Michigan, doing a research and trying to teach students the classes.
Right. Okay. So you are used to teaching now. All right.
Yes.
All right. And then Kapish, how was your beginning with tissue engineering?
Okay, so the first encounter I had was during a family gathering. So
Ah.
one of our relative got a kind of a bypass surgery, and they were discussing about that thing. So one of the over enthusiastic person in the group, he was exaggerating things and saying like that in ancient times we had techniques that we can replace the whole vessel with a lotus stem, something like that.
Right. Right.
And I was like intrigued by this, that like what is this he is saying. And at the same time we had internet at our home, so I tried to go to the internet and try to look for it. I couldn't find any reference for what that person was saying, but I did find references of I got introduced to this idea of tissue engineering. So that was back in
Yeah.
2006. I was in my 10th standard, but I was introduced that okay, it is possible that somehow you can use these artificially or lab-made organs, and there is an idea to put them back into the humans.
Right.
And my interest peaked in 2008. So in January, around January 2008, there was this. I'm not sure if you remember. There was this thing in the news as well. Doris Taylor Hart. So she's a
Yeah.
professor in Minnesota, and what she showed was that if you take a heart, you decellularize it, and then you put the cells, it can beat after 8 to 9 days.
Yeah.
And when I read about it and when I saw it, really fascinated me. Means that was the one of the thing which actually motivated me to take biotechnology and then biomedical engineering. So I was introduced to this through just a curiosity of after listening that okay, such things can happen, and then seeing that a 2 minute video of that beating heart.
Right. I can imagine that must would have been very fascinating at that age, at least.
Yeah.
Uh, I'm sure we all get fascinated even now, not just in that
Yes.
age.
Yep.
It's very interesting that way. And I think so. I think Nilesh had a presentation that he wanted to make. Let's
Yes.
begin
I
with that.
can start sharing that.
All right. Okay. So it's a combined presentation. All right.
Yes.
Yes.
1 second.
As we thought, our reviewers are going to be general people also, so it will be good
Right.
for them to have the overview. What really tissue engineering
Of
means.
course.
So just to have a basic idea, I think Dr. Gupta and we have made one small presentation, and
Right. Right. Yeah. It would be good to begin with an introductory session on tissue engineering before we continue with it.
Can you see my screen or not?
No, not yet. So on the share screen option at the bottom.
Yep, I'm trying to do that. Just give me 1 second.
Yeah, sure. Take your time.
Okay, I have to quit it. Just give me a minute because it's saying you have to quit it to share the screen.
Ah, okay. That's strange. Okay.
Yep. Okay.
All
okay,
right. It's all right. It doesn't matter.
because it's
Yeah,
it's, it's interesting.
we can continue while he comes back. So yeah, maybe I can continue with the things that I wanted to know of you from before he comes back with his presentation. So yeah, since it's I mean we have already got to know that you are dentist by training, and then you started to work in biomaterials thereafter. So all right, I think he's back. Yes, he's back. Oh, sorry. Yeah, sorry. So is everything okay, Kapish?
Yeah. Can
You can
you?
see your screen.
Yep.
Yeah. Okay.
So can you see the PPT?
Yes. Yes.
Yes.
Okay. Okay. So Nilesh can
Oh,
start with this.
so this is really interesting. We suffer from a lot of diseases, whether ranging from skin, which is the most commonly damaged thing, to bone, to CNS, which sometimes takes time, to heal and things like that. And we really wonder whether do we have a concept to build this. Before ancient time, doctors used to perform transplant, getting the grafts, but still those things are quite complicated. And then, can you do next slide, Kapish? And in 1997, we all might be familiar with this image of Prof. Vacanti's mouse. I'm very sure even people not working in the bioengineering or a tissue engineering, we have seen this thing. And this is what Dr. Vacanti has made, have grown an ear on the back of the mouse, and with the combining a principle. So that's what called tissue engineering principle, in which he combined the biology, he have used the cells part, and he have used the biomaterial to grow the cells. And this is the second image, a third year being done by one of the performance artists from Australia, Curtin University. And this professor, he really wanted people to hear his voice. So actually he grew the third year in his arm, and he have implanted the chips and the internet so he can talk with the people. Although these two have a very different perspective, but they share a common principle of tissue engineering, which is can grow a teeth, which is actually replication of a human developmental process. Till now, we have a regeneration like cell therapy and things like that, but still doesn't replicate the tissue human developmental process. And that's where the tissue engineering comes, where you can combine tissue engineering triads like scaffolds, materials, and the growth factors to really guide through that human developmental process. And from now, I think Kapish can take and finish the presentation.
Yeah. So again, this image has also like growing the ear on the arm or the nose on the arm is also being recently been used in some of the U.S. Army personnel, because sometime during the war these extremities get disfigured. So they have been trying to use this to make a kind of a synthetic ear or a nose which can be replaced for aesthetic purposes, and something which you see also over here. So, just to give you a brief background about what the tissue engineering encompasses. So, it is basically three main components. The one is the cell. So, that depends on the specialized organ that you are creating. For example, if you are creating a bone, you will try to work with osteoblast. If you are working with heart, you will try cardiomyocytes. And there are then supporting cells. So these are the cells which support the organ. And again, we can discuss it in detail later, but there are a lot of different cell sources like adult cells, stem cells, and recently a hot topic, induced pluripotent stem cells, which we refer to as iPSCs. In addition to that, you also need to provide these cells some factors, mechanical factors, as well as growth factors. So these are the chemical compounds which stimulate the cells to function in a specific way. And the other thing that we require is a scaffold. So scaffold is basically a matrix which supports the cells. So this is the image of that decellularized heart which I was talking about. So even if you remove the cell, we have a kind of a skeleton or a scaffold which supports the cell. And this is something we need to provide, which provides shape as well as functionality to the organ. So in summary, we can say that the overall process or the dream process of this tissue engineering will be that we take the cells, we expand them or culture them, we put them with certain material or scaffold, let them grow in, with the growth factors so that they start functioning as an adult organ, and then we transplant it back into the human. So just to give you a brief background of how this biometal can interact with the tissue, the one method could be just a passive interaction. For example, like a suture. So it is just there to hold the tissue. It's mostly inert, and its function is it doesn't have any biological function except for sealing the thing. The second could be an integration function. So here, if you see this image on the top, you see that the tree and the building has been integrated. You can still differentiate that what is a tree and what is the building, but they have integrated so well together that it functions as a same unit. And in the below, you see then the picture where you see a biomaterial as well as bone which have integrated together with each other. And the third kind of biomaterial is which supports the cell, but with time it gets reabsorbed and is replaced by the normal material which is secreted by the body. And in this case, we can define the material into three aspects. One could be conductive, which basically allows some cell to come into replace the tissue, or inductive, which basically promote one cell to come and don't allow other cells. And the third is the cell transplantation, where we seed cells along with the material. So in this sense, a scaffold has basically three main functions. One is to act as a carrier, that is for the cells to be delivered. Second is to act as a barrier. This is for the unwanted cells, because we generally do not want the immune cells to attack the scaffold. And third is as a reactor to allow the cells to differentiate and to transport. And again, just to give you an example, that we have been able to have some successes in designing some simple structures. But what you see over here is a structure of the skin. So there have been advances in past 30 years where we have tried to develop certain materials which can be used to replace the skin in case of burn injury or some other wound. So if you will see some of the basic constructs, these don't have any biological component in them. They just have a sealant layer such as a silicone as well as a matrix which is the collagen. But with time, there have been the development of some new tissue engineered graft which also have cells which can provide a better support to the skin. But in all these cases, even with this, the the graph that you get is nowhere near the skin. For example, what you see over here, this hair follicles, none of them available method has been able to regrow this hair follicle. So the skin that we generally use it as a tissue engineered skin can have a barrier function, but it cannot produce sweat, and it doesn't really have any hair follicles. And just to introduce some of the other applications, we have applications of lab-grown meat and drug testing, where we try to miniaturize the organs in microfluidic devices for the drug testing. And just to give you an overall, what does this is compromised of? This is compromised of a lot of different fields. You need cell and molecular biologists to design cells. You need immunologists to see if there will be any immunological effect. You need electrical, chemical, and mechanical engineer to devise the devices, surgeons to implant, and policy makers. Because with all this, there comes a plethora of ethical issues. So you need policy makers so that such policies could be implemented, that a fair use of these materials can be done. And again, you need material scientists like Nilesh to be able to design the materials which are bioactive and which can have some biological applications. So over to you, Nilesh.
So that's what the tissue engineering, the last slide which Dr. Gupta presented, that is very important. Before, we used to work as, okay, I'm a cell molecular biologist. Oh, I'm an engineer. I'm a policy maker. I'm a surgeon. And that used to be very tough. It's very tough to get something out of it. And now with the advancement in the science, people are working together. As you can see, being a clinician, I come to research. So I have that perspective of, okay, how we really use these products in clinic. And sometime I used to work in a company with a stem cell banking, and patient used to ask, doctor, when I can use my stem cell, things like that. And we have made a very big progress in this situation where, policymakers are really helping, because there is a potential here in FDA. I think 2017, they have made a new guideline for a tissue engineer product, because they are not considered as a biological product or a medical device. Basically, tissue engineering is a combination. So although we as a researcher, we can do a lot of stuff, but in the end, it is a policy maker. They are the one who can really going to make these things come into the clinic. So that is also one facet of this tissue engineering, which is really important, when we really want this products or the things to get to the patients.
Right. I'm glad you talked about all those aspects possible for any kind of a tissue engineer. But I would like to take you back to biomaterials first, because that's one of the first things that you would think about when you are preparing any engineered tissue. And then there's always a concern about tissue engineering in general, that most of the things that we have been working on, that's still in a very nascent stage. I mean, most of them have not been able to actually make it to the market or to the real life implications. So Nilesh, maybe from you, I would like to get some insights, because you are a dentist by training. Also, you are doing the research on the material side. So as far as biomaterials goes, that how much of it do you see being used in the real life solutions?
See, in the real life solution, I will give a very simple example. Being a dentist, dentistry is all about materials. You go
Right.
to a doctor's, you go for the amalgam filling. It
Yeah.
is simply the amalgam, it is like a metal. Simply, if
Yeah.
you go for a composite filling, the tooth colored filling, it is again a biomaterial. And, biomaterial is something which we are using in healthcare, for long. If we give the example of old dentistry, people used to use the gold and the wood also, to build the tooth and make the tooth to hold together. But we have progressed a lot, honestly. And as being a collaboration, before the material scientists, they used to work only on the material to make, now we have seen the advancement from a TV. It is a part of a material. We used to have such a big TV. Now we have a flat screen TV. It is actually
Right.
the advancement, and that's the engineering perspective have come back into the medicine. And medicine people talking with the engineering, and clinician talking with the person, and they come together. And I was talking a few days back with one of the cranioplastic surgeon, and I asked him, what do you really want, being a researcher. And I was asking him, like, what you want, basically from a thing which can make your patient's life easy. And he says, like, elastomeric sutures. And
Huh.
that is something we as a researcher sometimes we cannot think, because whenever there is an injury, it gets swell. And when we
Yeah.
suture, sometimes the suture doesn't hold that thing.
Right.
So that is very important for us to talk. And this is something which we talk with the clinician. We get to know, how we can advance this biomaterial. And with
Yeah.
the advancement of a nanomaterials, before it was a
Yeah.
bio, now we have seen a lot of nanotechnology coming. And
Yeah.
again, if I have to give an example, sorry, pardon me if I'm giving a lot of dentistry example,
No, no,
because
no, no.
that
I'm
is
glad you are using the real life examples.
because in clinics, patient comes and we have this implants coating is something, as Dr. Gupta talked about, the integration, some material they need to integrate. And in dentistry, the dental implant is something which really have to integrate, and that is something which patients are using rather than using a removable implant, so that, when they go to marriage, they have to take out and they cannot eat. And now the
Yeah.
implant has come, and that's the all because of the advancement in the biomaterial. And we have this coatings, and sometimes the coatings which are thick, and they result into the corrosion of the implant and the infection of the implants. And with the advancement of these nanomaterials, I think we are managing to reduce this thickness, which can result into less fractures of this coating, so that implant can live longer. Even for the stents, we have a shelf life. After 10 years, maybe we have to go and replace, because although the things are not corrosive in biological environment, although in the
Yeah.
lab, but when we put in the body, it is a very complex system. It's going to corrode. So we are getting with the coating materials, exactly the nanomaterials, if I talk like that. So they are really progressing things. And when we talk about the organ printings, now this is something which is coming up again because of the advancement in the biomaterials. We are really seeing things progressing, which can result into really high resolution of things. And so even like, we want a material which can, change shape. So actually,
Yeah.
material scientists are working on a things, so when they put in the body, so if there is a swelling, so it can expand accordingly. And that's where the hydrogels or some polymers combination. So now in the medicine, we have a lot of composite material, a combination of a biomaterial and a combination of a hydrogel. So I think we have pretty much advanced. And being a clinician and people working together, I think the biomaterial is something which is going to really make this tissue engineering field go ahead, because we have a knowledge about cells and the growth factors. But
Yeah.
the for a tissue engineering, the biomaterial is something which we still not very sure about how to use those things, because depending upon the system, it changes properties. And we have known, I will give you one real example. It's been work done here in Michigan in 2014, and they have implanted this scaffold in patient. Actually, that was supposed to, it was a PCL scaffold, actually, a biomaterial, and it's supposed to degrade in 3 months. But when you put in patient, and it was more bulkier, and instead of 3 months, it didn't degrade it in 2 years. So,
Oh,
so it is something which we really need to think, how we can make this biomaterial really advance this field of tissue engineering.
right. I'm so biomaterials is one of the most. I mean, as I mentioned, and in all the examples as Nilesh has been citing, biomaterials is one of the most important things that you would need to think about when you are actually designing a tissue. So that I mean, this is something that already Nilesh mentioned, the 3D bioprinting technology, and this is one of the most sought after technology in the field of tissue engineering currently. And I mean, this is something that I've been thinking about lately, is that how do you see that most of these are being used in the research labs right now. So since both of you are involved in this research, how do you see 3D bioprinting actually coming down to the usage in actually making the live organs which could be used for let's say implantations? Uh, Either of you could take this question, or both of you could answer this based on your experiences. That how far have you seen the organ printing, organ printing out of 3D bioprinters being realized in real life implantations?
Kapish?
Let's say, okay, Kapish, yeah,
Yeah.
yeah. Let's begin with you
So
first.
uh, one of the So let me talk about that where bioprinting can help us a lot.
Yeah.
So when we are thinking about designing a organ or something of that sort, one of the most important aspects is the vascularization.
Right.
So normally what happens is that in any structure, it needs oxygen and to be supplied. And for any vascular structure, the approximate distance that from any vessel the oxygen can travel is around approximately 200 micron. So you need within any structure at every 200 or so micron a vascular structure which can provide oxygen at that level. Now, this has been one of the major challenge in the tissue engineering because if you see all the advances that we have, so mostly the advances have been in bone, like bone implants, either a skin graft, urinary bladder, or some vessels. Most of them are very less vascularized organs that you don't need that much vascularization because they are either a single layer or a couple of layer cells, or in case of bone implant, when you put it, you don't need that much vascularization, and that has been one of the biggest impeding factor in the development, that we are not able to provide the correct vascularization. But what can be achieved with the 3D printing? Because again, that depends on the resolution of the printer. The last I remembered, we can go up to a single cell resolution that we can print at a single cell around 30 micron droplet size. So that's basically your how much one pixel is printed in the printer. Maybe Nilesh is using something even better. So it provides us a possibility that you can integrate, you can generate this design, the blood flow design or the vascular design within the structure. So that is the one of the advantages it has as compared to other methods, because the other methods where you let the cells grow, where you let the cells populate a scaffold, there you do not have that freedom to actually regulate or engineer or compartmentalize, in a say, the various fractions of say one compartment which is a vascular compartment, and second is the tissue compartment. Say if you're say generating a liver, you need hepatocytes or liver cells. You also need blood vessels, and you also need bile ducts. So you need all these two or three compartments. And the spatial localization is something which can be achieved very easily in principle with 3D printing.
Right.
Yeah.
No, no, as Dr. Gupta mentioned, 2011 was the time I saw Prof. Anthony Atala in a TED talk. He has that, a kidney, if I remember correctly, in a live talk. And that was quite fascinating, actually. And that time he said, that is the field which is going to really advance. And I was back in India at that time working for this company. And that really motivated me to do a PhD. And so I think
Right.
I took a sequential, I learned a biomaterial, and now actually I'm glad to say that I'm doing a bioprinting. So that is after doing a bioprinting, I can realize, as Dr. Gupta mentioned, the vascularization and the resolution is a big challenge. Although we have some success stories, people have printed some liver patches. The biggest company, the Organova, so they are the
Mhm.
one who is really pushing these things to go, and clinicians from the Rice University, and they have all those success stories. They have a beating heart, but as Dr. Gupta mentioned, getting a heart is a very complex structure, any human organ, and we can get the vascularization at the outer part, but getting the
Right.
vascularization deep into the tissues, is something which is very challenging, and that's where the resolution of the printers come. And I think since 2011 till now, we have made a good advancement in that, having this printers which can really increase the resolutions, and they can put those micron scale. It is not only the circulatory system, because the organs, they have their own circulatory systems also, which as we progress they develop by themselves, so that is something which is a very challenging part with the bioprinting. Although we
Right.
have some success story, for example, people have transplanted, like, trachea and things like that, but things with that, we have some setbacks also with that, but that's how we learn. Before we were not able to implant, but now we are getting this complex geometries and things. The other part with the bioprinting is the advancement in the bio ink. you know that is something which really plays important role, how or what kind of tissue we can print, because if I'm using a polymer, basically, So I'm not sure that, for example, if I use a PCL, it is hydrophobic, or it might give us a very good resolution, but it is hydrophobic, and it might not really help into the tissue integration kind of thing, although getting a compartment, things like that. But that's how the hydrogels and things have come. And with the bioprinting now, we can place the cells, so we can have a core and shell bioprinting actually, so you can have a tubular structure, as Dr. Gupta was mentioning, inside, and outside then we can have the cells covering, so they can form the tissue outside,
Yeah.
and the cells they can grow. So core and shell, they have that is another technology which have advanced, and nowadays the printers, they come with the multiple print heads, so if you want to print a structure or a support, with a polymer, so that to get a hard part, and the hydrogel can be used together to get a soft part. And there are a droplets on demand printing, so if you want to place a single cell, at a specific location, so we have that capability also now, so we can print these things in a single cell, and we can have the cells placed, accordingly, and the places we want. So I think the printer has advanced, and surely I think if the way we are working, and all the policy makers, scientists, researchers working together, I think this is the hot topic. And I will say this is a fourth pillar of tissue engineering, as we saw, like, a biomaterial and things. So this is called a biofabrication. I will say a biofabrication, basically, not a 3D printing, and additive manufacturing, or a biofabrication, which can really make this tissue engineering field go to a next level, where I think we
Yeah.
can address a lot of challenges which a normal tissue engineering have faced. And I think it's something which is very exciting.
So another thing that we were discussing earlier, I mean, I think you mentioned it in your slides already, is something that people keep talking about, called stem cells, and very popular names in terms of uh, saving the stem cell, cord stem cells, particularly, uh, uh, especially for stem cell therapy, generally, which is still, to some extent, seems to be in future. But I mean, it has been somehow stayed in a research environment for a very long time. But for some reason or the other, the actual usage of stem cell for disease solving has not become as popular at it could have been, of course. But in general, but anyway, things have changed quite a bit ever since the development of genetic engineering also. So like we could make iPSCs at some point, thanks to reverse engineering the differentiated cells, or if for simple understanding, let's say something like a skin cell could be de-differentiated into a stem cell. How have you guys been using genetic engineering for your purposes? I would ask that question first, and then I'll get back to some usage of stem cells that you guys are doing. Yeah. So let's go for Kapish first.
Okay, so I'll come to this iPSC part.
Yeah.
So the one of the major challenges with the iPSC field is still that yes, we can get a cell which is a stem-like cell. But the main question is, can we get back a cell which is an adult functional cell from it?
Right.
So I have worked a lot in the liver field, so I can talk from that perspective. So most of the time, almost all the available protocols give you a cell which is more or less like a fetal hepatocyte. That means it is the hepatocyte is basically the main functional cell in the liver. It is you can go as far as a hepatocyte which is there in a newborn baby. You can't get an adult hepatocyte as of now. So now why does this matter? Say, for example, if you have 100 cells and they are doing some function of the liver, say production of albumin, and say 100 cells in a normal adult produce 100 microgram of this albumin, just for an
Right.
example. But the cells you can get, if you take 100 cells, they will only produce 1 or 5 microgram. That is like 5 or 10%. Do they actually have then that this capacity to be able to be used in any way possible? So that's the one question, that yes, we are able to get the differentiated cell, but in most of the cases, and this I'm talking about again, so there are simple organs like skin, blood vessels. Yes, you can get a pretty mature adult cells, from iPSs. But for most of the complex organs, such as liver, heart, the main problem is that it is still we still don't know how to fully differentiate them back into the cells which are similar to the adult cells. That's the one thing which has impaired most of the work which is being done. Because in our lab we tried to use some IPSs to even generate some structures, liver structures in lab. But again, the expression level was even less than from human iPSCs was even less than the hepatocytes that you get from rat or mice. So that is one of the main challenge that still need to be addressed. That how do you develop methodologies to get the cells as at par as the functionality of an adult cell. The second question which has impaired the usage is uh, that these cells, any biological system is a very noisy system. So this is
Right.
generally which happens with any system. And however less, there is always a small probability that these cells can in turn give rise to cancerous tissue when they are injected in the body. So that is the another thing which has impeded or which has stalled the actual usage of these cells, the stem cell therapy in any clinical application. Although I like to say that there are a lot of places, like if you go to Middle East or Europe, there is this fancy term of stem cell tourism, where people inject claim that they can cure any disease by injecting stem cells. But as far as I know, there is no actual technique which has been approved which can treat any of this disease yet. The only thing which has come up in the recent time is this CRISPR technology.
Right.
So initially there were also some, because we have to be cautiously optimistic about these techniques, because in past these techniques have failed. So yes, people were optimistic, but they were also cautious about this technique. And then there were some reports that okay, that yes, they can have some off-target effects. But it is only recently, I think it happened in February 2020, just before this corona crisis started, that the first clinical trial showed the safety of the first genetically engineered T cells in cancer patients. Again, what the group was able to achieve, that group, I think that group was also in the University of Pennsylvania, that group was able to show that at least these genetically modified cells are safer in the patient. The efficacy is still debatable because even after 9 months, the cancer was still growing. But it has gone to some part that okay, we are going into the field where we have some safe technologies which can be then later used to develop some other stem cell based technologies. So I think in the recent time, the one of the most promising technology which has come up is this CRISPR-Cas technology, and the second technology which has come up in the recent time is this the reinvention of this blastocyst complementation technology. So again, everyone might have heard about this ethical considerations of the using embryos or blastocysts, because what you are doing is basically you are taking the cell from an embryo and you are destroying an embryo. But with the advent of this iPSs, it was figured out that instead of using an embryo, you can use these iPSs and then use transplant into the blastocyst, and then that can develop into an organ in that organism. So you can have basically a rat which is growing a liver which was from the iPSC derived from mice. So they basically that is growing the mice. A rat's mice is growing in the liver, or a liver's mice is growing in a rat. But with this again, there was this ethical consideration that again people saw that some of these in some of these cases, these cells can be, because you are basically doing the manipulation at the blastocyst level. So you are taking the blastocyst, taking out some cells and putting some iPSs, which can grow into certain organs. That there were some leakages of these cells into two main things. One is the gametes, and the second is the neuronal cells. And this is where the major ethical consideration is, and that is where, as you talked earlier, the policymakers comes into picture. Because if we assume that in future it might be possible of taking some human iPSs and to grow those cells into in an animal, or to grow that organ in an animal, then we don't want these two things, or ethically these two things will be bad. One, if they start producing human gametes, And second, if they start getting incorporated into the brain, because then that would mean theoretically that animal will have an intelligence as far as a human, theoretically. So again, this thing was started in 2012 and 13, and then again it hit a roadblock because of this, because it was almost banned almost everywhere. All the government agencies banned it. But then people said that okay, what they did, and this is again a recent study, I think in 2019, where what they did, they took the iPSC, they knocked down the two genes, one which is required for fertility, and the second which is required for neuronal development. And then they showed that okay, the cells, the organ which are grown from these genes will cannot develop into either gametes or neuronal cells. So again, this whole field has been buzzing with the possibility that this can be used. So yes, there are all these techniques, but again, all these techniques are at a very nascent stage, and there are a lot of ethical consideration, policy consideration, which need to be tackled out before they can be used in any clinical settings.
Right. So yes, I think Kapish speaks a lot from the research point of view. Maybe Nilesh has a different point of view when it comes to usage of stem cells in real life implications. Maybe yes. Over to you, Nilesh.
I agree with a lot of points which Dr. Gupta mentioned. It is genetic engineering is something, it's being there for a long time and people have been doing, but there have been some setbacks. And
Yes.
actually, I remember one movie here, The Rampage. I know everybody knows The Rock from the WWE. So he
Yeah.
has that movie Rampage. And when that movie starts, they show this gorilla is being genetically engineered using CRISPR. Okay. And as he mentioned, you can make somebody as capable as a person with a human capability, as Dr. Gupta mentioned, theoretically. So actually in that movie they show that actually theoretically a people who the gorilla which grows and he has an emotion, he gets anger and things like that. So this is something, that's where sometimes people come and talk us, given the current situation of COVID, where is the vaccine? And I really like the post which you have posted, when was the last we followed the vaccine development,
Yeah.
never before.
I'm glad you read that.
I read that, it's a very good insight. I think everybody should read that. That's where we come. How when we follow and we see the technology and especially the newspapers, they say, oh, this being done here, this being done here, and then they come to a clinicians, and they ask, why can't you do that? As Dr. Gupta mentioned, this is at a nascent stage. We cannot do that things, and I have a first-hand experience using the iPSC, and
Yeah.
although we have done all the karyotyping so that to know, the cells are healthy and they cannot lead to tumor. And we have implanted that in mice. And, we got all the good mice, but we found one tumor
Uh-huh.
in one mice, although the cells were sorted and everything nicely, but one cell is good enough to destroy. And that's how we have seen a lot of implants nowadays being called back, because they have some things like that. So that's why,
Yeah.
the restriction is very important. And, people, who are really reading things, they really need to understand this technology, there needs to be regulated very well. And we have to start simply. In my lab I use microRNA. It's not like a genetic engineering things like that, but it can really increase the expression. So I can use a specific microRNA if I want to make a bone. So maybe with the 3D printing scaffolds I can just have this microRNA coated and just put,
Right.
so it cannot change much of the things. And with the genetic engineering, it is very important for us, to look, I'm trying to treat a bone disease, but it's a gene, and body can function in a multiple way. If I have to give you a simple example like ALP protein, we see
Hmm.
an increase in this ALP protein when we are suffering from liver cirrhosis or even liver disease. But
Yeah.
ALP is one of the common marker again when we do a bone formation. So it is very important for as a researcher and a people, to really understand, because when we do something in the petri dish, it's a very controlled environment. And when we talk about something putting this into a human or using it for a clinical trial, it is a very complex system. Even we study medicine for 5 years, 6 years, and when patient comes, it's still we debate ourselves, we discuss with our colleagues, what is the approach we have to use. So same it,
Yeah.
in the research, it is that's what we have to discuss. And people around me, they really need to be careful, when somebody talking, as Dr. Gupta mentioned, the stem cell tourism. And in India also, it is very common. India and Mexico is actually the hub for stem cell tourism. And the people, they really need to understand, sometimes they just give you a cocktail, things like
Hmm.
that, PRP, things like that. It has a stroma cells. Basically, it has all the growth factors and cytokinin kinds, which can really result into the good results, maybe, but we really need to be careful when we talk about the genetic engineering. We can see in that movie, they have a big wolf, and actually in that movie they show they are doing that research in space, because they were so afraid, because if something happens, it's going to change the humanity. And that's how it happens. It comes to the earth and this infect these three creatures, and then how they fought. And it's a beautifully made movie using the science of a CRISPR technology. So I will recommend people to watch that. What really happens, if you don't have the control of this research technological,
Yeah.
so
Yeah. A lot of biohackers are actually would actually disagree with you. That
I would like to add a point also
yes,
here about
yes.
so like he said that in India also, I personally know of a relative who has a lung fibrosis, and uh, there was this one quack doctor who said that okay, I can treat it with injecting iPSC cells. I'll take the cells, make it into stem cell, and then treat it. So I'll just like to share you one example which happened, I think, in 2010 or 11. So this was a clinical study when this iPSC field was gaining traction and stem cell field was gaining traction. So they thought that, okay, maybe we can take the iPSCs, inject into a person who has had an heart attack, and then this cell will replace the cell and form the cardiomyocyte, and the heart will again start beating. So this was a clinical trial, and when they injected it, what happened was those cells turned into bone cells, bone-like cells. Now the reason was that when a when a heart attack happens, there is a instant fibrosis at that in that region, which basically changes the mechanics of the tissue. And when
Right.
you put a cell which in such a hard environment, it sees a bone-like environment and can differentiate into a bone-like cell. So that's why the material, the mechanical properties, and the situation you are dealing with, it makes you have to understand all of that before going into any such treatment. It is not possible to just put the cells, because if something has happened and there is a scarring has happened in internal tissue, that will be a very hard tissue, that will be a very stiff tissue, and the cells will behave in a completely different way. A same stem cell which on a soft substrate can give you an adipose tissue, on a hard surface can give you a bone like tissue, and you don't want a bone in your heart, means that's the last thing that you want.
Yeah.
So for any of these techniques, it has to be fairly well regulated. Means I know that the regulations can be pain, but for most of these techniques, the regulations has to be there to prevent any misuse and any damage to the people who are receiving it.
Right. I'm glad you spoke about the mechanics involved in the
but we
relevance of
have
mechanics in tissue engineering. Yes. Sorry,
we
Nilesh.
no, we have a success story even with the stem cell transplant recently.
Right.
I know the group personally in Japan, and
Uh-huh.
they have done this. It's published in 2017 actually, and that's the first group to use a dental pulp stem cells, to really do the the pulp regeneration. So we don't
Right.
have to go for the root canal, and
Right.
that's quite a success story. They have done a follow up of 6 months, but again with the stem cell technology, that is something we really have to do a 10-year follow up
Of
actually.
course. Yes.
And,
Yes.
and recently, the some of the here in United States, they have approved a clinical trial actually to
Mm-hmm.
use the stem cell, and it's a multi-center trial happening in United States and the other part of the world actually. So that's good that people from US, Europe, India, they are collaborating and really using this potential, so that your question of, patients are banking the stem cells, so how it's going to go. So we as a researcher, we really working hard, to
Right.
really make this come long. And when we talk about a cord blood, the cord blood is actually being used,
Yeah.
but when
Yeah.
we talk about the tissue, is a different thing, because it can form anything. As Dr. Gupta was
Right.
mentioned, we don't want a bone in the heart, so we need to
Right.
be really careful. But with the cord blood, it is a hematopoietic component, so it forms the platelets and the RBCs and things things like that. But we have some success stories, and as a researcher, everybody is working hard to really get this tissue, the mesenchymal stem cells, if I have to say, into a
Yeah.
clinic. So fingers crossed, we will have more positive news in coming times, I hope.
Yeah, I'm glad that I conquer. I mean, you, or rather, my answer conquers with your answer. That's because that's the answer that I give to people when they normally ask me if they should be saving their child's cord blood cells, uh, and then this is the answer that I give them, that maybe 20 years down the lane, technology would have changed considerably.
It is, it
So
is, it is like an insurance. We pay the
yeah,
insurance, right? So, like,
right,
we don't know when we gonna get it. And I know my parents have done the LIC for me, and
right,
now I'm big, but I touch wood, like, we don't suffer. So it's all faith.
of
And
course,
and
yeah.
research is a field. We have a faith. In any field. It's all about a faith. So that's how it is.
Right. So there's another topic. I mean, since we have spoken so much about stem cells and their usages, there's another thing in the field of tissue engineering, which has been a very hot topic for a very long time. That is the organ on a chip, or in general their body on the chip, if it's that possible. And I know that Kapish, your PhD lab and even your postdoctoral lab, both of them have been working on one on the liver on the chip and another one on bile duct on a chip. And then how so I know that these things, when you can develop them, or rather when it is developed properly, they can be used for drug testing. I know many of these organoids which are being developed are being used for drug testing, even for coronavirus. uh, I mean, the drugs against coronavirus. So we see the real life usage of these kind of chips now. How do you guys how do you guys develop it?
Yes.
Second, how do you see the usage in drug? Because I think, I mean, sorry, in drug testing. So because I
Okay.
know this is one of your dreams also.
Yes. So first, like I like to say that I think that this organ on a chip is a fancy name for one of the technique for culturing cells. That's it.
Right. Yeah.
So basically, we need to culture cells. We culture cells outside the body, so we can test a lot of things. And there are many cultures. I think the audience would have known that they culture cells in petri dishes, like on a flat substrate. We culture cells in a kind of gels so that they can grow into some 3D organs. And in this, we culture them, the cells in small devices. Now, as the name says, organ on a chip. So it doesn't mean that we have a miniaturized heart or miniaturized lung. But what it means is that, so let's see what first an organ is. Organ is composed of multiple tissues, which are composed of a variety of cells. For example, in a liver, we can have liver cells, which are hepatocytes. We can have blood cells. We can have endothelial cells. We can have immune cells. And all these cells, they compose this organ, which we call as a liver. Now, as I said, organ on a chip is a culture system, but it has two major advantages. One, that it uses microfabrication, and the second, it uses microaerofluidics. And both of these things adds a kind of dimension which are not available with any other culture system. So what microfabrication adds is compartmentalization as well as providing of special cues. So what does this compartmentalization means is that you can have small compartments where you can put different kinds of cells which can interact without mixing up. So this is the advantage which compartmentalization provides. And microfluidics provides the ability to flow the certain things as well as interconnectivity. So now, with this background, let us see how does a drug works, and then we can come on to what would be an ideal human on a chip kind of a device. So, for example, if you take any drug, the first thing it happens is that it is dissolved in our body, in our stomach, and it is absorbed through our gastrointestinal tract. From there, it goes into the liver, and liver is like a black box. So what liver does, it metabolizes anything and everything you give it to it. So it can do, so it's a black box because we don't know what will be the end product. It may be a beneficial product or it may be a completely toxic product as well in some cases. Now that the product is then again secreted into the, probably into the blood, and it will go into all the organs like our kidney, heart, brain, everywhere. So what we desire from a drug is that first it should have safety, that it should not do any damage to all those small all those organs. The second is efficacy, that it should be effective at the organ it is intended to. So for example, if I have a drug which is say to change the beating of the heart, so it should go, it or its metabolized product, when it go to kidney or brain, it should not do anything. But when it goes to heart, it should change the beating pattern. So that is something
Right.
that we desire from a drug. Now, what we can do with this microfluidic is that we can create this small compartment where we can replicate certain functional aspects of those organs. For example, what we require from a gut, that there should be an adsorption. So we can have a layer of cells in a fluidic chamber where the drug can be adsorbed. And from that adsorbed channel, we can send it to a small other fluidic device which has a liver cells which are metabolizing in a similar way as an adult liver will do. As I said, most of the cell system doesn't do that, which is a limitation. But again, the idea is to get a liver compartment where there are like 100 to 200 cells which can metabolize the drug as a liver would do, and then whatever is the product, it can be sent to all those smaller other organ systems, or basically the cell system like kidney, and see if they are not doing any damage to that. And if we have a kind of a system where we have provided a kind of a few cardiomyocytes, it can go there and we can test if it is changing the phenotype of those cells or not. Now, what this whole process does is that it reduces our time of testing, because the only other way to test whole this thing is either to test in animals, that you give the drug to an animal and then you see how the whole process is going on, which one is very costly, and again it's very time consuming, and in most
Right.
of the cases it doesn't replicate the result. And the second thing will be, which will be completely unethical, to if you have 1,000 drugs to test, you put some 10,000 people and give each one of them those drugs and do that, which is again not feasible. So to cut that part, what we do is what the ideal system would be, that which have cells grown in these individual chips which are interconnected, and where we can test the drug as from different compartments. Now, this is the ideal world, but we are very far from it. So currently, what we have is we have miniaturized versions of some of the organs which replicate one or two functionality of the system. Say, for example, let me take the example of this HCV virus or any other virus. So with this, what we want to see is if it can replicate in a cell system or not. So any fluidic device or any system which I make, my only goal will be to see if that virus can replicate in that system or not. So we are at this time at that point where we can replicate certain aspects, but not completely mimic all the functional aspects. Now, coming to the part which my lab is currently working. So let me just give you a background. So there's the bile duct on a chip. Now, bile duct is the duct where from where the bile flows into the gallbladder and then again into the intestine. Now, there are a lot of diseases which attack this bile duct. And if it is damaged, you will have jaundice. And if the bile flows, bile is very toxic. It can lead to death as well. And we don't have any good system to study it, because it is a very you can see unique compartmentalization, that one side we have a very toxic bile and the outside we have a kind of an ECM and blood and everything flowing on, and these don't generally don't mix, these both compartments, and it is very difficult to recapitulate this compartment. But as I said, what we get with organ on a chip is compartmentalization. So we can, in that we can make the compartments kind of a tubular compartment where the cells will grow, and an outside compartment where we can provide the correct the matrix signals. And in that, what we were able to form was a small tubule around 150 micron in diameter, 1 centimeter in length, which was in certain aspects similar to the bile duct. Now, how we can use now this drug? So again, to give you an example, there is a disease known as biliary atresia. So it affects the neonatal children, and what it we still don't know how it what is the cause of it. Is it genetical? It is environmental, or God knows. The only study that we have is from an Australian livestock, where we saw where people observed that whenever there is a drought, the incidence of this thing increased, and they linked it to probably a kind of a plant that the cattle graze upon. So other than that, we don't have any knowledge that how does this disease happen. But now with this duct on a chip, we can test a lot of compound to see if any of those compound induces this damage. And once we have the induced that damage, then we can incorporate other test other compounds which we can then use to see if we can rescue that damage. And this is something would be a big advancement from animal testing, which generally takes around a month to test one drug to see if the duct is being damaged. So that's how it reduces that time, as well as it can be further used for a personalized medicine if the iPSC technology extends so far that we can isolate the cells and make the person specific bile duct or a duct or an organ on a chip and test the drug. So that's again a very futuristic thing. But for now, most of these devices are we are trying to replicate some functionality of a disease and then to use that to study how to treat or how to screen for various compounds to treat those diseases.
Right. I'm glad you had you brought all the perspectives of the future drug testing in this answer, because of course, as you mentioned, you cannot go and test the drug on 10,000 people.
Yes,
Testing a drug on 10,000 people is a very bad idea. Uh, but then there is another success story of tissue engineering so far, at least in terms of startups. I would say is the lab grown meat, or another name for it is cell based agriculture. Uh,
yes,
and this is turning out to be quite good for most of the startups, because they can produce meat for at least for animal already. Human meat is, I mean, for human, the meat edible for human, sorry.
yes, yes,
uh, the meat edible for human is still under testing for most of them, but I know there are several of them which are already trying to make burgers out of it. So it's turning out to be quite good. But so Nilesh, I'll begin with you first. How do you see the biomaterials in that case playing the role? And then Kapish, I'll come to you about how the integration of tissue works in that case. Yeah. So
I
Nilesh,
think
you first.
it's a very interesting field, as you know, nowadays we have a lot of animal lovers, so I think, and people, they want to turn vegan, but at the
Right.
same time they want to have that feeling of a meat and things like that, so we already have in the market, the plant-based burgers and things like that. But this thing is definitely going to change things, and a lot of big companies actually, they are putting money, and I know there's big research going in Australia,
Right.
especially with this, and even Google have some things doing with this. They have also started funding with this. Talking about the biomaterial, it is not that complex compared to when we talk about the human organs for the tissue engineering. It is a simple
Mm-hmm.
process of taking a cells from the animals and just growing on a scaffold, and these scaffolds are, basically is a gelatin. They are basically a biopolymer, and
Right.
gelatin is something which is already approved, and this is something which we eat, the jellies which we take. I think many people here in the United States, they always keep chewing on something, so that is something which is made up of gelatin, and that is safe for human consumption also, I think. But the main challenge comes is growing the cells on them and really getting those bulky meat, because we want to look for the industrial scale up, so we really need to have this. Maybe I'm not sure very much about this, but people might be looking to modify this materials, so they can really, when the cells are growing and when you are expanding this, making this as a good meat, not just as a single layer of a tissue, or maybe processing to break them. So, so still some modification needs to be done when it comes from a material perspective. When it's good to grow, material on the gelatin, and obviously
Right.
they gonna grow. But when we have to really expand them and make them into full grown tissue, it's gonna take a lot of challenge, and the cells, they're just not gonna grow on that, So how they're gonna have those extracellular matrix form? So when we talk about biomaterial, that is simply as an extracellular matrix, so maybe, not gelatin, maybe having some more lipids or polysaccharides kind of thing. We know the cellulose which we have from the plant, that is something which can really help making this thing streak. But I don't have much say on this with the biomaterial perspective, I will say. But those mostly is going to be, the gelatin, the cellulose, those kind of thing, which is already approved and safe for human consumption. So that
Yeah.
is something, is the most commonly used. And gelatin, as I mentioned, is something which can be used very nicely, things like that. So that's, I think, with here, we don't have to play much with the biomaterial, but
Hmm.
the main part comes from a tissue engineering perspective to get the bulky meats.
Right, right. So Kapish, your insights on what kind of cells are being used in these processes, and are they edible when they are not
Okay.
when they are still in the lab? First of all.
So, okay. Yes, there has been basically a lab grown burger, means beef which has been made into a burger. But
Yeah.
let me come from a perspective, from a application, from a research perspective.
Right.
So what happened was that for almost all these years, people have been trying to make this tissue engineered products in the lab. So in the lab, you have a very good reactor where you can pump all the nutrients, oxygen, and everything. But as soon as you take them out, they are basically dead meat because they
Yeah.
don't survive. And
Yeah.
that gave the idea of this, that anyhow, if it is just after taking it out, it's a dead meat, then why not use it for consumption? So, so the idea is basically, because we are so inefficient in making a real organ, we can just make an organ which is inefficient and can probably replace the beef industry, because this is a part. This comes from a part of our inefficiency to actually develop a functional organ. We are developing a dead organ. It's basically a dead tissue. But the main question which comes here is that and which that there was this a very beautiful kind of an there was a company which had made this meat, and they called people to test it out. The main problem is of texture.
Right.
That I'm myself a vegetarian, but I don't know much about eating meat. But what came out of that whole discussion was that yes, it may have the same nutritional value as a normal meat would have. Okay, it may be able to have the similar taste, but the texture, which is basically differs from what a good meat is and what from a bad meat is, that is something which is still not, even in this dead meat, we are still not able to fully understand that how to produce that texture. And in that, I think a material science, and especially the microfabrication and the nanofabrication, can come into play a role, because yes, the
Right.
texture is okay. It's very difficult to tell what the texture is, but it comes from, you can say, the way the meat is cut or the surface characteristics of the meat. And so when it is tiered, the how does the surface characteristics of the meat appear to be? And that, I think, is one place where again, texture is very difficult to define in terms, or in some, at least for now, in terms of engineer terms. So this is something that how do you feel something, like the feel of a thing. But this is one thing where I think the topographies, nanotopographies, or the microtopographies, or even the material engineering can come into place to provide that texture to these tissues. And there have been some advances. Mostly, again, this will be okay. The most of the big companies are basically trying to. There is a French name, I can't pronounce it correctly. It's called Fulgra or something like that.
Foie gras. Foie
Yeah,
gras.
I can't
Yeah.
pronounce it. So
The
basically, it's
company's
a
name is Gourmet.
basically what it is. It's a fatty liver.
Yeah.
Okay, so
Duck's
normally when
fatty
you,
liver.
sorry,
It's a duck's fatty liver.
yes, it's a duck's fatty liver.
Yeah.
So
Yeah.
basically, when you want to, so the people who say they eat meat for the protein. Okay. But the only thing which has gained traction is fatty liver. So basically, no one wants to, means from a nutritional standpoint, people don't consider meat as a source of that fat. They consider it more as a source of a protein.
Hmm.
And the reason is that because inducing fat into the liver is very easy. You just add any lipid, it will become fatty in itself. So that is again a kind of an application which has come up because of our inability to grow proper muscle cells in vitro.
Right.
like to provide the texture of the muscle cells. So that has also come up from kind of that inability to grow a proper muscular meaty organ. So yes, this is a very yes, people are working on this, and I think that this can lead to a big industry in future, because yes, people are trying to go away from the non-veg, and again with this coming of this coronavirus which can come from animals, people, it might happen that people try to move away from these safer options of the meat, because again a lab grown meat, it's very difficult to transmit any virus, because you have treated it with almost though there is the safest condition you can give to a grown meat. But the two things that we need to that are major challenges is, one, how to provide the correct texture so that it feels good to the person who is eating it, and second would be to actually use the vast knowledge of the muscle tissue engineering into generating the meat which is actually nutritional to the body also.
Right.
Again, we have to apply all those knowledge to give a functional meat, not a functional, but a meat which can be of a similar texture and similar quality as a normal meat. And the third issue which will come will be the cost, because there have been instances where the lab grown meats have been made, but they are normally, even the cheapest which I have read was around 5 times more costly than the normal meat that you get in the market. So yes, maybe some of the rich people might prefer that meat, but again, if you want this thing to go to a lower level of the society, or to the major part of the society, you will have to think of the methods to decrease its cost.
Right. I have only one point to add to that, to the
Yeah.
texture, lack of texture that you were explaining. So that also comes from the fact that there's not proper integration of each kind of different cells that are being used to make this meat, and
Yes.
definitely there is a lack of proper biomaterial that is being used. So at this point, maybe we can start taking some questions, since we have almost crossed 1 hour and 12 minutes. We'll just take few questions. So let's go for this question, because we were talking about it. Kapish, would you consider an artificial meat to be vegan? I mean, this lab grown meat to be vegan meat. Would you
Okay.
eat that yourself?
This was a discussion I was having. Okay, so and my question to that person was, if I take human cells and grow a humanized meat, would you, means he's a non, would you be able to eat that meat? So this is a very ethical dilemma for people, because anyhow the initial cells are from animal, and probably even like Nilesh mentioned about gelatin, which is a scaffold. Gelatin also comes from the cow's bone, like the
Yeah,
collagen comes from the cow's bone, which is then denatured into. So in, yes, in a theoretical aspect, it will be vegan, but will people accept it? That's a different question, and that will be, I think, very personal to everyone. But yes,
right.
it can be made, except for the cells part, which again you just take some small skin cells without killing the animal. Most of the rest of the process could be vegan, because right now what is being, again, the most important thing which is being tried is to use plant-based additives into the meat, so that the entire meat is grown with the additives which are from the plant. Except for the cells, I think everything else will be plant-based.
Yeah,
So again, the terminology is for the policy makers to decide, but in terms of eating, it will be a personal choice of everyone, that would he be able to eat or not. Like it's with the egg. You call egg as an unfertilized egg, but some people consider it vegetarian. Some people again consider it as a part of non-veg.
and
so
and
it's a debatable topic.
it's
Yes.
a very debatable topic, Shurvi, and this is again, like, it is a personal perspective, as Dr. Gupta
Definitely.
mentioned. We cannot say a word about it is the vegan or not, but
Yeah,
I can say one thing, if it is not tasting like a normal meat, and
right.
here in the United States, like, people say if the meat is not juicy, so it is
Yeah,
definitely a vegan, so that is a personal choice, and people, they have to decide whether it is a vegan or not.
yeah, definitely. I think it's the same case with the plant-based meat. I mean,
Yep.
a lot of people like it, a lot of people don't. So yes, let's go to the next question. So the second question is about that this person wants to know. how to grow skin, artificial skin in the lab. So, or like maybe, yeah, you can take it in either ways. Yes.
Nilesh, you want to take it?
Yeah, it's like the skin and the bone is something which is already people are growing, as Dr. Kapish gave the examples, already the derma graft and all those things. They are like the lab grown, and I know a very good friend of mine personally. He do this skin, he grows the skin in the lab. So it is pretty much possible to grow skin in the lab. And but if I have to say from a dental perspective, when we talk about oral mucosa, it is a
Hmm.
lining again of a mouth. It's like in
Yeah.
skin, but it is a very different when you have to grow the oral mucosa, because a lot of blood vessels involved compared to the normal
Yeah.
skin part and things like that. So it's a very different game. But when we talk about skin as a general, I think it is pretty much possible, and we already have tissue engineer products into the markets, which
Right.
are lab grown. So
Right. Yeah, I would just want
So
to add to that that there are a lot of cosmetic companies which are developing skin for testing cosmetic products. Yeah. All right,
and
the
Kapish.
only
a lot of the skin grown products are used for a drug testing for the cosmetic
hmm,
product, as you mentioned. And I have a very good friend, and they have done this, and there are companies using their skin to do that. Even they have those perfusion and things like that. So it is
Right.
pretty much possible. Yeah.
Right.
I would just like to add one thing to it. The only problem with the current artificial skin is that there are no oil glands and no hair on that. So that's the one again, one of the biggest challenge, or the challenge we have is that how to incorporate those things into that system.
Yeah. Yeah. We are close to perfect, but not perfect yet.
Yeah.
Yeah. So the next question is that what would be the industrial potential of biofabrication? That question would be for Nilesh, I suppose.
That is something we are seeing like every day. There are so many startups. I know back home, like the IITs. They have a lot of good success stories. And giving the if I have to talk from the Indian perspective, so governments are funding for the startups. I can see a lot of people coming out from IITs and even from institutions. They are getting well supported. I think that is one industry which is really, last year was like the year of a chemistry and physics, but this is the era of a biofabrication, and
Yeah.
it incorporates, this decade is going to be the biofabrication decade, and a lot of industries coming up. We are seeing, the as the last slide of Dr. Gupta presentation, the biomolecular scientists, engineers, and everything, So, and that's how we are improving. And every day we are seeing, this bioprinter or this printer or maybe this technology is better, So I think the potential for this biofabrication industry, I think it's quite great.
Right. So we'll take the last question now. I will shorten down this question maybe. Is that I think so? So the all right. Maybe let me read it. In the field of running science suggests that gene type plays an important role in the athlete's performance. Athletes who are generally in the strength and endurance sports like long distance running. I want some information regarding the gene effect with athletic performance. Kapish, would you want to take that?
This is something beyond my expertise. Sorry. Yeah, I don't
Since
know
you are an
about.
athlete and you know a little bit of genes.
No, this is something which
I can put some point in this,
Yeah,
yeah,
sure.
like again, I will say this is a personal perspective and how motivated you are. And I
Right.
know Kapish, he was not a runner at all, okay, before, but now he runs 8 to 10 kilometer every day, even he has done some running. So I think, like, the genes, they definitely play the role, but there is a field of an epigenetics, sometimes,
Yes.
a twin born, in United States, and when I put that one twin in India, although born to the same mother, there is definitely going to be a change in the complexion. There is going to be the change in a lot of things. So that's why it is a genetic, but there is something called epigenetic field, and I know my brother-in-law is working on this things. So how you train your body and how you have grown things. So you definitely can change that perspective. Even Gupta, as I give example, I'm being a runner, but now I have slowed down, because I'm lazy. But now when I
Hmm.
talk with Kapish, he's running 10 miles every day. So it is
Yeah.
again comes from a motivation perspective. So it's like, that's how we can say.
Right. All right.
So
So maybe we.
one thing we
Sorry,
can,
Kapish.
yeah, one thing I can just add to it is that uh, this key that okay, does the cell play a role or the environment play a role? It is like from a macro to a microscopic level, because you don't know that it's a chicken and egg problem, because sometimes the same cell with the same material, you give it a different environment, it will behave differently. So there was this one study, I think it was either muscular dystrophy or progeria. I don't remember the context, but it was a. So in both of the cases, what happens is that your muscles start weakening very early on in the life.
Mhm.
Okay, and muscular. You must have seen that PA film from Amitabh Bachchan where they show that early aging. So it was one of those conditions. But what they did was they took two mice, they put one on a normal cage and one they put it on a running mill. And after the 9 months, the one who was on that running mill was completely normal, even after the genetic defect. So that,
Right.
and the one who was kept in a normal again progressed with the normal case of disease. So sometimes you don't know. Yes, genetics has a very important role. It has a very important role. But there are some things where the environmental factor or how you uh, there are these environmental factors which can help persons overcome those genetic limitations if there are any. Yeah.
All right. There is one question from Hemant for you, Kapish.
Okay.
What are the commercial opportunities for bud tissue engineering in India? I think Nilesh, you also know Hemant, so both of you can answer these questions.
So in India, one thing is that the again because from Hemant, maxillofacial surgery is the one of the most like sought out surgery, because we have a lot of what you can say a lot of accidents, and the first thing that gets hit is this region. And the maxillofacial surgery is something that okay, in other parts my arm is there, I can put a structure and I don't care much about the shape, that how does it look. But with face, everyone cares that okay, I should look good, and this is a very aesthetic concept as well. So now recently, like again, there have been things that okay, what used to happen was that they will take out bone from somewhere and fit in over here or over here, and that will give you a very disfigured kind of a face. That will keep you alive, but that will give you a very kind of a disfigured kind of a face. But with this tissue engineering, the techniques have come up. For example, recently there was I was reading somewhere, there is this BMP laden some matrix which is put into this maxillofacial part, and I think this was also approved as a clinical trial. It can regenerate the whole region, and the whole region can then give rise to a very normal like shape. So what you do is that you basically so you scan the face. Say if the person has a defect on one side, you take the rest of the side, you use mirror that to engineer a kind of a structure which can fit in over there, so that your shape doesn't disfigure. Put some cues such as a BMP and other things, put it back, and then they have seen that in some cases it has been regenerating something similar to normal. So, and this is something which happens a lot in India, and this is again just one example. So there is a huge demand for such an industry, but the only thing is how well the reach of this system would be, because these systems are very costly.
Right.
So right, and this is something which again I might be considered sadist in this case. I think it's good that this is not in everybody's reach, because now only the rich people can be the guinea pigs for this technology. So these techniques are available for because as Nilesh said, you need to see for 10 to 15 years that how this technology turn up. So now initially the rich people can be the guinea pigs, and we can start India, and this technology can be introduced in India. And over the time, when the technology again, as the time progresses, if it is working, it will become cheaper. The patents will expire, and all these things will happen, and then it can percolate into the general public. But for now, I think there are a lot of applications, but again, there are some policy changes also which are needed in the country, as well as the cost part need to be taken very seriously, because some of these maxillofacial implants have shown very good results.
Right. So,
Nilesh?
Nilesh, how would you answer this question?
No, I think Dr. Gupta covered most of the part. This is something, again, it depends upon educating the people, And it's like, this is more from a industrial marketing perspective. If a company, how good you are can convince the doctors. So it is not something, being a clinician back home, some people, they come and they just tell me, okay, this is the product, you use it, okay. But sometimes the clinicians back home also were aware about the dental stem cells. So the company, they really took the time. I was working for the company doing the CME and things like, educating the dentist, who are the first contact when I talk about the dental stem cells, and the gynecologists are the first contact when we talk about a cord blood. And if they don't tell to the patient, they are not aware of these things. And it really depends upon how the people are marketing and how convincing the reports are. And
Right.
as we are seeing, the governments are doing a lot of changes in the policy making. As I mentioned, in 2017 here in FDA, they have revised the tissue engineering products. So I think if we have a lot of good success stories now, as I will not say like guinea pigs are only the people. I have seen a lot of people who cannot afford money, and this is something, the company, sometimes they can fund, to do some of the surgeries as is being done here in the United States. If there are some things, as we know, and it depends, it's a part of the government also, the regulations, how they regulate and how they can see these things happening. So I think it is a lot of potential. Example, the best example is the maxillofacial tissues, because integrating those tissue engineer product in the maxillofacial is not a problem, because it is a full of a blood supply. So the chances of necrosis when happens in this is pretty less when I talk about a skin, because it has a lot of blood supply. Even with the one cut, we can see a lot of bleeding. When I do a shaving, I get a cut, I see a big bleeding. But I do something here, it doesn't happen. So I think it has a lot of potential, but it depends upon the companies and the person marketing those product. And definitely these have gone through a GMP lab setting, and all those things is important.
Right. So there's one thing that I would want to add to that, there are thanks to 3D bioprinting, there are few companies which have begun to work in India on implantations, generally either for eye or for liver, because these are slightly more manageable organs in terms of making them. So yeah, I mean, even that direction is quite uh, fastly developing field generally all over the world, and even in India, there are few companies already working in that direction. And I don't think it would be very far-fetched in future to see a cell-based agriculture company working in India as well, because there's a lot of demand on that side as well. Maybe considering India is a partly vegetarian country, so maybe it would be more on plant
I,
plant
I,
tissue culture than animal tissue culture. But
I don't think so. India is a partly, there are more people non-vegetarian in India back another part of the country. But I would like
yeah,
to add one more thing with the integration of artificial intelligence.
yeah,
So that is something, which is again changing, as we were talking about the stem cells. So even getting one population of the stem cells. So, the artificial intelligence is something which can reduce these errors, and they
hmm,
can really help in the advancement of the field, because the quality testing of this product is very important, and a human testing. And if we have, like, I know in the clinics nowadays we are doing this, using this augmented reality. Now a lot of
right,
video games also, we are talking about this augmented reality. So for
right,
example, if in a clinics I'm doing a procedure, okay, so
hmm,
that's how now in our time, our professor used to check. But now
hmm,
with the current artificial intelligence technology, so if I'm doing some procedure in my pre-clinics, not on patient, in the pre-clinics, so that's how we are getting trained. So we have this thing which tell us, okay, you have done the procedure correctly. So it gives us the light, a green color light. So the procedure was done correctly. So we can use this artificial intelligence also, and it is quite a advancing field. I can see now people doing coding at the age of 7. There are courses
yeah,
in India also, people doing coding at the age of 7, and we don't know the spelling of coding. Maybe I'm sure when I'm 7 year old. So I think that's how the field have advanced, and this integration of artificial intelligence is also something which is going to really play. So you can increase the manufacturing, the product testing, and knowing the reliability of the products. Using this
yeah,
integration. So definitely, and India is, I think, is a booming economy given the state. I see a lot of bad news, but I don't think so. A lot of companies like now when we talk about the academics. So now we have a lot of spin outs coming from the academics, even the industry. So there is a good collaboration between industry, academics, and even the hospitals. So I
Right.
don't see it is a potential, but as we know how well you in the market and how well you are, so I think that is something which is going to play again a big role.
Yeah, right. Thanks both of you for all these beautiful insights. This is like high time for us to stop now.
I agree.
1.5 hours of talking on tissue engineering. So, so yes,
Can I add one thing?
yes, sure,
Yeah,
yes,
so we were talking about vegan meat. So there was this one talk about vegan organs. So
right,
what they were doing was that they were taking plants, leaves, decellularizing it, and then recellularizing it with things, and then they were saying that
right,
okay, this can now function as a blood flow. I just remembered that the blood flow pattern of the nerves. So basically, the
uh-huh.
plant
Veins,
the veins can be used as a vessels, and they
right.
they were testing that idea. So that might be a vegan organ for very strict vegetarians to transplant.
Maybe that would be one way to convince vegetarians to have meat.
Yes.
Agree.
Yes. Let's see if you would want to be a guinea pig of that. But all
Yeah.
right. Thank you both of you for joining us today, actually.
Thank
And
you.
yes, so let's stop it now. Thank you very much.
Yep.
Thank you very much.
Thanks.
Bye bye.
Bye bye.